Okay, so we're going to also do a full lecture on nervous tissue, the central nervous system, the peripheral nervous system. Basically, most of April is going to be spent in neurology, in special senses. And then at the very end, we will do endocrinology, and that'll be the last thing that we do. So the next few weeks, we'll be talking about the nervous system. So I'll actually film this and get it up. Or get it up onto YouTube so you guys can access this on Canvas. Okay, so let's talk about the nervous system. This is just a fascinating area. Let me see if these guys are out. He's getting messages from Bernie and his friend. We're here. We just don't want to come in and bug you, but I don't see them out there, so I guess they're not here. They're just probably late. And, oh yeah, we're really here. Okay. All right. So we need to. If they come in late, we really need to shame them. We need to shame them very good. We need to look at them. Okay, look at them. We need to make them do the walk of shame. That would warm my heart so much. Yes. All right. So let's talk about the nervous system. This is fascinating. It's fascinating because for a couple of different reasons. Fascinating, one, because we don't fully understand what's going on. That's the first thing. We don't fully understand it. We don't have a robust theory of consciousness. You guys have probably all taken psychology, right? Most of you are looking at taking a psych, because if you're looking at like nursing school or something like that, you'll probably have to take a psych. And you talk about consciousness, and we all say, well, consciousness is the awareness of yourself and the environment. Well, what does that mean, though? It's really a fascinating thing to think about. What really is consciousness? We just don't quite have it pinned down what exactly it is. But what we do know is that it appears to be some sort of property, possibly a property that emerges from complex systems of cells in our nervous system. How about that, huh? So when we look at the brain, okay, when you look at your nervous system, your brain, your spinal cord, you have about 100 million functional units. And what is the functional unit of the nervous system, this functional cellular unit? Say it again. The neuron. Yes, the neuron is the functional unit. We have, on the order of magnitude, about 100 million. Okay, so I want you to think about this. From 100 million cells, okay, and then each of those cells have, on the order of magnitude, of about 20,000 connections. Okay, so you've got 100 million neurons interconnecting and interweaving, each of them about 20,000 times. So how many connections is that? That's a lot, right? On the order of magnitude of 100 million times 20,000. And I want to put this in context really quick. There are about 100 million stars in the entire galaxy that we live in. The entire Milky Way galaxy, about 100,000 light years across. Okay? So if you could travel at the speed of light, it'd take you 100,000 years to cross the universe. Okay? Just imagine that. And there are about 100 million stars. So there are as many neurons in our brains as there are stars in our galaxy. Okay? And then when you think about the connections, individual connections or synapses, you've got multiple galaxies worth of connections. So just think about that. That's an amazing, incredible thing to think about. So what we're going to do is we're going to talk about some of the functional or foundational cells that we find within the nervous system. The first thing that we need to understand is that the nervous system can be broadly separated into two areas. You have the area that exists within the axial skeletal system, and that's known as the central nervous system, or the CNS. Okay, the CNS or central nervous system. And then you have all of the nerves that branch away from the central nervous system, and they go into the appendicular, okay, And what are they known as? What is that branch known? Say it again. The peripheral, right? Periphery. So you've got the central nervous system and the peripheral nervous system. And we will talk about those in much more detail next week. Okay. So far so good? You guys okay with that? So you got your CNS and your PNS. And the reason that I want to differentiate that so early is there are different cells in the nervous tissue of the CNS versus the PNS. There are some slight differences. So let's now talk about the foundational unit. So the functional unit is known as the neuron, and I've drawn this, and this is pretty similar to what you guys have in your lab manual, kind of. Yeah. Okay. And so what we need to do is we need to appreciate some of the parts of the neuron, but let's look at some of the other cells. So you have the neuron, this functional unit, but that functional unit can't do its job without a significant amount of support. Okay? And so you actually have many more of these support cells than you do neurons. And you have several different types of support cells. And collectively, what do we call support cells in general? The glial cells, the neuroglia. Okay? So the neuroglia or the glial cells are the support cells. So you have glial cells and then you have neurons. So let's talk about some of these support cells. I've got the neuron here. So I've got this cell right here, and it has these little projections, okay, that kind of wrap around this part here, this part of the neuron. They wrap around, and they have little hand-like extensions that attach to the vascular supply. Okay, so they kind of anchor. They're really good at holding, protecting, and anchoring the neuron down. And what are these known as? Those are known as astrocytes. You guys okay with that? Astrocytes. So I'll just write that here. Find a marker that a little more robust. So astrocytes. You guys okay with that? Now, we also have cells, little spindly looking cells, very small, that basically surround the neuron here. And they have little spindly like connections. And what are those known as? They're small. Small, kind of surround the neuron. We're not quite sure what they do. There, say it again. Glia, there you go. The microglial cells. Yeah, so you've got your astrocytes, you've got your microglia. All right. It's these yellows. I've kind of tried to color code it the best I can. Okay. Now, we also have cells that line cavities, the insides of cavities within the central nervous system. And what are these cells known as? They are ciliated. Right, they're ciliated. Say it again. The ependymal cells. Good. And what do the ependymal cells do? What's their primary function? They produce something. They secrete something. They secrete a kind of fluid known as cerebral spinal fluid, CSF. So the brain is so vital that it has double protection. Like in every other organ, the brain is covered, okay, in a layer of connective tissue that protects it, known as the meninges, and we'll talk about those in more detail. But then you have these inner areas, these openings, okay, canals, and what we call ventricles inside of the brain that are fluid filled. And then you have fluid that surrounds the brain itself underneath the meninges, or underneath, okay, the protective lining. So the brain is not only surrounded by a protective lining, but it kind of floats in a layer of cerebral spinal fluid, right? So it's kind of a double protective mechanism. And it is these ependymal cells that secrete and then circulate the cerebral spinal fluid around. You guys okay with that? Okay. Now, we also have another type of cell. And this type of cell, well, all of these cells are more specific to the CNS, but this type of cell produces a sheath or a coating that covers part of the neuron. And it's a kind of insulating coating. And what are these cells known as? Well, let's make sure we get our terminology right. What are the cells that make this protective coating known as in the central nervous system? The oligodendrocytes. And this is central nervous system only. In the peripheral nervous system, okay, it's not the oligodendrocyte that makes the protective or the insulator. It is, in fact, the Schwann cell or Schwann cells. And another thing that you need to know is one oligodendrocyte can produce multiple myelin sheaths in the CNS, but in the peripheral nervous system, one Schwann cell, one myelin sheath. You guys okay with that? Okay, so there are some differences between CNS and PNS, and that's a big one there. Okay.
So Schwann cells are in PNS?
Yeah, Schwann cells in the peripheral nervous system. Oligodendrocytes in the CNS, yeah. CNS only, PNS only, yeah. And then as you correctly identified, this is known as the myelin sheath. You guys good so far? So, what we have here, the way that the nervous system works in general is there basically comes down to three concepts, okay? Sensory information needs to come in, okay? So sensory information needs to be brought into the nervous system. The nervous system integrates or makes some sort of sense of that sensory information, and then it sends out, okay, motor information. For example, if I grab something that's really hot, sensory information about that temperature potentially burning my hand goes into my central nervous system, and then my central nervous system sends out motor information that tells me to, you better let go of that, dummy. Right? That is the basic process going on, okay, with the central nervous system. So sensory information, sight, sound, hearing, balance, right, what we call proprioception, okay, smells, pain, pressure, heat, or temperature, okay, all that information, okay, comes in, your brain integrates that, and then it sends out motor information. It reacts to what the signals, the information that comes in. You guys okay with that? That's in general what happens. All right. So how do these neurons, what do these neurons do? Well, neurons, as we alluded to during the musculoskeletal system, they are capable of transmitting signals by depolarizing. You guys okay with that? And when a neuron depolarizes, there is a wave of depolarization that starts in the body of the neuron and then makes its way out away from the neuron, and that is known as an action potential. When that wave of depolarization moves away from the neuron, it's known as an action potential. You guys okay with that? Now, what we need to understand is that there are three major parts to most neurons, and we'll talk about different types. Okay? So you have these extensions here that bring information into the neuron. Okay? And what brings information into the neuron? The dendrites. Typically, the dendrites. Okay? So information comes into the neuron, and then information can leave the neuron, and information leaves the neuron via what kind of structure? The axon. Good. And then where the nucleus is, and most of your organelles, the smooth and rough ER, the mitochondria and all that, what is that known as, this round structure here? This is known as the cell body, or we also call it the soma. Soma means body, okay? You might see either term used, the soma or the body. So far, so good? Okay, so in general, how does it work? Well, what we see, and so what I've done is I've enlarged here. I've enlarged the soma. Okay, so here I have the body, and I have this area right here next to where the axon comes out away from the cell, right? So axon goes away, dendrites goes toward. And this area here is known as the axon hillock. The axon hillock. Okay. And what we have is, we have the cells in a polarized state, like muscles. Which means that where is most of the sodium? Outside, right? So the sodium-potassium pump pumps the sodium out and keeps the potassium in. So you see where I've drawn this here, right? Most of the sodium's on the outside, most of the potassium's in. So when we look at the inside of the neuron, the inside of the neuron compared to outside is negatively charged. Does that make sense? Because most of the positive is outside. And that when the neuron is not doing anything, okay, that's what we call the resting potential. And the resting potential is right around negative 70 millivolts. And I've drawn a little graph for you guys here. The potential versus time. Okay? So this is the resting potential. Are you guys okay with that? It's at rest, nothing's happening. Okay, the sodium's out, potassium's in. Sodium potassium pump's working. And then what happens is information starts coming into the neuron. And information can only do one of two things to a neuron. It can either stimulate the neuron to depolarize or inhibit the neuron from depolarizing. That's it. Okay, so you have excitatory stimuli and inhibitory stimuli. Okay, it may be pain, it may be swelling, it may be some neurotransmitter, okay, it may be light, okay, whatever it may be, it'll either stimulate or inhibit the neuron. You guys good with that? And so what we get is, let's say that right here, I have some information coming in that stimulates. Okay? Stimulates the dendrite, and that information comes in. A stimulatory stimulus will cause sodium channels to open in the dendrite, and it'll allow a little bit of sodium into the cell. Okay? You guys okay with that? It'll allow a little bit of sodium. So when I have a stimulus that stimulates, okay, the cell, I get a little bump. It goes up a little bit. You guys okay with that? All right. When I get another stimulus, because remember, you've got like 20,000 connections, right? Some of the information coming in is inhibitory, some of it's stimulating. So maybe over here I have inhibitory information coming into that neuron. Okay? And generally, inhibitory information causes chloride ions to move into the cell. And are chloride ions going to make the cell more or less positive? Less positive. So there was that stimulatory, and then now I have an inhibitory that drops it down a little bit. You guys see that? Okay? So I'm having this happen, you know, all this information coming in. And these are making tiny little changes in the membrane potential, okay? These tiny little changes that don't result in full-on depolarization are known as graded potentials. I believe your lab manual uses the term local potential, okay? But graded potential tends to be the common term that we use. A graded potential. So, what ultimately has to happen? Well, what has to happen is, do you remember we go back to the Hillock here, right? There are sodium channels embedded there that are voltage gated. These are voltage gated sodium channels. And so the membrane potential has to be high enough. Once it gets high enough, it triggers these voltage gated channels to open, and then the entire neuron will depolarize. Okay? And the voltage at which the neuron depolarizes is known as the threshold potential. So far, so good? So if you cannot get up to the threshold, these voltage-gated sodium channels will not open, the neuron won't depolarize, and it won't send information out. You guys okay with that? Does that kind of make sense there? And so ultimately what it is it is the sum of all the inhibitory and stimulating information coming into the neuron, all of the impulses that inhibit it or stimulate it, okay? And we call that temporal summation, okay? And when the sum of all the inhibitory and stimulatory impulses are enough to let enough sodium into the neuron to reach that threshold, and this is about -50 here, but once you get up to that threshold, you have enough stimulatory impulses coming in. Once you hit that threshold, these channels open up, and the sodium rushes into the neuron. And once you have sodium rushing in, it depolarizes the neuron, and then you have what's called an action potential. And then the neuron, the sodium-potassium pumps, reset the neuron back down to the resting potential. You guys okay with that? But it will not depolarize if you cannot get above the threshold. Okay? You guys cool with that? So once that threshold happens, okay, sodium starts rushing into the neuron. And then the axon begins to depolarize. But here's the thing. The reason that we have myelin wrapping the neuron, many neurons, the axons, is the same reason that we have insulators wrapping electrical wires, right? You have a wire that you plug your computer into, right? That wire is surrounded by an insulator. What would happen if it wasn't surrounded by an insulator? What's that? Yeah, all the electrons moving out of there would kind of have a tendency to leak out, right? Same thing with the neuron, right? All that sodium, okay, would kind of have a tendency to kind of get dispersed and leak out a little bit. So you insulate, right, that, and that allows for improved conduction of that action potential. And then what we have is we have these little spaces in between the myelin sheaths. And these little spaces are known as the nodes of Ranvier. R-A-N-V-I-E-R, Ranvier, okay? And so what happens is you get a special kind of conduction pattern known as saltatory conduction. Okay, saltatory conduction. Okay, it's kind of like hopscotch. So what you get is you get a lot of sodium ions concentrated here, right? They're really concentrated as they rush in. And then the sodium ions start diffusing out and they move away from each other, right? And by the time they get to this node of Ranvier, they're really spread out and dilute. And so you have sodium channels in the node of Ranvier that open up, allow more sodium to come in, and then they amplify that, and then that signal goes back through, kind of spreads out a little bit, and then it comes to another node, more sodium comes in, amplifies it, and then so on and so forth. Okay? Until that impulse gets to the end of the axon, or what we call the axon terminal. All right? And in the axon terminal, we have vesicles filled with chemicals known as what are the chemicals in the vesicles? The axons known as? Say it again? Neurotransmitters. Good. All right, and these neurotransmitters are released out into this space here, and this is known as the synapse. All right, and these are neurotransmitters. And these neurotransmitters will then attach to receptors on the dendrites of another neuron, or maybe some junction with a muscle or gland. Okay. And then these neurotransmitters are either going to be inhibitory, they're going to inhibit that other tissue that they are attaching to, or they will stimulate it. Does that make sense? All right. You guys okay with that? And what we say is that this neuron here is the presynaptic neuron before the synapse, and this neuron over here is the postsynaptic neuron, or after the synapse. You guys cool with that? Okay, that's all the detail that we need to go into for today. So really the rest of the lab is going to be you guys getting microscopes for your tables. Okay, there are 3 different types of slides. I've got nerve endings, I've got teased out nerve fibers, so you can look for those nodes of Ranvier, and then spinal cord with neuromuscular junctions. So please pull those slides out, make sure you're comfortable with them, make sure that you can identify a node of Ranvier. The nerve fiber, the teased nerve fiber is going to be your best bet for finding one of those nodes of Ranvier. Make sure you can identify the neurons and the glial cells. You will not have to identify individual glial cells for this class, but just glial cells versus neurons.